Now we're gonna look onto the genetic codon table. That is a codon table for both DNA and RNA. What is the difference in both the tables? So if you see for adenine, which is present in the DNA, is replaced by an uracil molecule in the RNA. So only those compounds will get differentiated. That is a thymine is replaced by uracil instead of adenine. So now we're gonna see about the codon table for both DNA and RNA. If you take in DNA, the thymine has been replaced by the uracil molecule in the RNA. So in the table, only those difference will be seen. So I'll give an example for your table. So this is your first, I'm going to take you to the RNA table. You can see this is UCAG, that is uracil, cytosine, adenine and guanine. And the same way you can see UCAG, the first letters and the second letters. So it is already indicated the first and second letter for your easy understanding. So here you have U, C, A and G. So the first letter is U. So it is gonna be the U. The second letter also it is U, because this table you have both first and second letters are U. And followed by U into U. Then you can write the same sequences here. U, C, A, G, U, C, A, G, U, C, A, G. And if you look at the thing, the first two amino acids or the first two codons will code for one single amino acid. So different amino acids will be coded by different codon. So this is a very important codon table. And similar way, if you're gonna write it for a DNA molecule, this is an RNA codon table. If a similar way, if you're gonna write it for DNA, then U should be replaced with A. So you'll have ACAG and ACAG. So TCAG and TCAG. So the U is replaced by T. So this is a codon table. You can see the first letter and the second letter denotion, and all other terms are like UCAG repeated. So once you write the table, and each codon will code for a different amino acid. So you can see the amino acid, the codons are specific for single amino acids. So one amino acid will be produced by more than one codon. That's why we call it as a specific thing. So one the process is known as, like if you take, this is an RNA codon table. If you go for your DNA codon table, the thymine is replaced by the uracil. So you can have TCAG and TCAG. So similar way, if you multiply and write the table, you will have the same amino acids which has been produced. The amino acids remains the same, only the U will be replaced by the thymine nucleotide. And if you see, there are 21 different amino acids, and you will have a stop codon, and that is known as occur, amper and opal. So this codons, here it is TAA, instead of U it is given as thymine molecule. So this is how your genetic code looks when after for RNA as well as for DNA. Now we're going to look into the role of amino acid tRNA synthetase enzyme. So this is an enzyme which is present in your tRNA. What happens is this amino acid tRNA synthetase enzyme will have an active site. So this active site will recognize a suitable amino acid and it gets joined to the active site. So once the amino acid is joined, that is amino acid's carboxyl group will join to the amino acid tRNA synthetase enzyme. So once they join through a covalent bond, an ATP energy is being required. So once the joining is taken place, after the utilization of the energy, a pyrophosphate molecule will be released out. So once a pyrophosphate molecule is released out, a suitable tRNA with a specific anticodon for that amino acid comes and binds to the amino acid tRNA synthetase enzyme. So once tRNA with the anticodon comes and binds to the amino acid tRNA synthetase, the remaining AMP, that is the 1-phosphate, is also released out. So it specifically binds to the 3-OH group of the tRNA molecule, binds to the specific amino acid. So this is how the molecule of tRNA becomes charged. So we are gonna see in detail this theory. So role of amino acid tRNA synthetase. So each, for example, if you are taking an amino acid, so if you take, this is as a amino acid tRNA synthetase enzyme, it recognizes an amino acid plus an ATP molecule, comes and binds to the active site of the molecule, that is the enzyme. So amino acid tRNA synthetase plus you will have an amino acid plus an energy, that is energy molecule ATP. Once they bind to the active site through the COOH bonding and covalent bond, a pyrophosphate molecule will be released out. Now, after the pyrophosphate molecule is released out, a tRNA molecule they'll recognize the tRNA synthetase enzyme. So once when the tRNA molecule comes and binds here, I am joining it here, 3', 5'. When the tRNA molecule comes and joins, it joins the 3'OH will join to the amino acid group. In that time, that is a specific anticodon. It is carrying a specific anticodon. This anticodon recognizes the sequences of the amino acid. So once a specific tRNA comes and binds to there, an EMP will be released. That is, the remaining phosphate is also released. That time it is termed as charged tRNA. So what is a major role of your amino acid tRNA synthetase is making your charged tRNA for the translation process. Second process is proofreading. It makes sure that only the specific amino acid comes and binds to the amino acid tRNA synthetase and hydrolyzes the incorrectly joined amino acid tRNA. So it is gonna go for a proofreading and it is gonna make your charged tRNA for the further translation process. And it is gonna undergo a proofreading, that is only specific amino acids will come and bind to the amino acid tRNA synthetase and hydrolyzes the incorrectly combined tRNA, that is amino acid tRNA, which is coming and binding to your amino acid tRNA synthetase enzyme. And this process will help, this both will help in the process of translation together. So the major function of your amino acid tRNA synthetase is the catalyzes the formation of charged tRNA. Catalyzes the formation of charged tRNA. So role of amino acid tRNA synthetase enzyme is catalyzing the formation of a charged tRNA molecule. If you take amino acid tRNA synthetase enzyme, they will have an active binding site where it is been recognized by an amino acid along with an ATP molecule where an energy is being required. So once it comes and binds to the active site, that is, once an amino acid comes and binds to the active site of the amino acid tRNA synthetase enzyme, the ATP is utilized and pyrophosphate will be released out. Now a particular tRNA will recognize this amino acid tRNA synthetase amino acid combination with the specific anticodon comes and binds here. The tRNA will have a 3' end and a 5' end. You can see in 3' end you will have your amino acid end. So this amino acid end comes and binds to the amino acid group. So it will produce amino acid tRNA will be prepared. So once when it is coming and binding, an AMP molecule will be released out. The energy which is utilized will be released out. This energy will make you the tRNA to be charged. So it is known as charged tRNA. This can be further used for translation process. Next, the major role of amino acid tRNA synthetase is proofreading. So once it is gonna make sure that the suitable amino acid is coming and binding to the active site. And the second one is, if there is incorrectly joined tRNA molecule, it is gonna hydrolyze it. That means it is gonna remove the incorrectly joined amino acid tRNA from the amino acid tRNA binding site. That is gonna make sure by this process. So the major function of amino acid tRNA synthetase is catalyzing the formation of your charged tRNA, which can be further used for translation process. Now we're gonna look on to the post-transcriptional modification of the post-transcriptional modification for an mRNA processing. So post especially for mRNA processing. See, this process is seen usually in eukaryotic organism. For example, if you take a prokaryotic organism, the transcription takes place in the cytoplasm itself. That means the DNA is being converted into RNA in the cytoplasm itself. That is, it becomes mature RNA, and the RNA will be transcribed into translated into a amino acid, that is a protein. Right. So what happens is, it is taking place in the cytoplasm. So there is no further modification is required. But if you go for an eukaryotic organism, the transcription takes place inside the nucleus. That means your genetic material, the DNA, is found inside the nucleus. So what happens is, if the transcription is taking place in the nucleus, the transcription will make a RNA. It is known as premature RNA because it is not in a complete process for the transcription to be completed. So what happens is, the premature RNA should undergo a maturation step so that it will be converted into RNA and RNA into a protein. So this maturation step, which takes place in the eukaryotic organism's nucleus, is termed into 3 different types. That's what we call as post-transcriptional modification. Before a transcription to be complete, this process has to be finished. The 3 processes of post-transcriptional modification are, the first one is poly A tail. So I'll go along the order. The first one is capping, right? The first one is, you're gonna cap your 5' end. Next one is, you're gonna do with the tail. So it is gonna go for polyadenylation. First the capping, and then you're gonna go with the tail. So polyadenylation. And the third one is splicing. So if you take a prokaryotic organism, the entire DNA will definitely code for a protein. There is no junk nucleotides which is present in your DNA. But if you go for an eukaryotic organism, they are made up of many non-coding sequences along with a coding sequence. That means your DNA will contain both coding and non-coding region. So if a transcription to be completed, this non-coding region has to be removed from the coding region. So only then the coding region will be joined together and will be converted into a suitable RNA. So now we are going to look into the detail of this process of post-transcriptional modification in eukaryotes. So I'm drawing it for. So if you take, this is your DNA, no junk nucleotides, so it is gonna code for RNA, transcribe into RNA, the RNA to a protein. Okay. But if you take here, you will have an extra material nucleus, and inside that you will have your DNA. DNA is made up of junk nucleotides. That is coding and non-coding. Coding are known as exons and non-codings are known as introns. So you will have a combination of exon, intron, exon, intron. So for this, introns has to be removed so that exons will join together. Then it will be transcribed into suitable RNA, then RNA to protein. So until this process, we call it as premature RNA, and it is present within the nucleus. Maturation takes place in cytosol. Cytosol. So the first process what we're going to look here is, capping. You can term it as 5' capping. So you will have 5' to 3' mRNA that has been produced within the nucleus. You're gonna cap it by the addition of, first enzyme is triphosphatase. Upon the addition of triphosphatase enzyme, it cleaves 1-phosphate group. So if you take the 5' it is actually made up of 3p. You will have your nitrogen base, then you will have your P, then your nitrogen. That is 3 phosphates will be there present in the 5' followed by a nucleotide sequence, that is a nitrogenous base with the phosphate bond to another nitrogenous base. Then in the 3' end, you will have an hydroxyl group. So what happens in 5' this 3 phosphate, when triphosphatase is acting upon that, 1 phosphate will be removed out. So it will be producing 5' to 3', you will have, then you will have your RNA molecule. Continuation, you will have an RNA molecule, right? Now what happens, the next enzyme which is going to act on this is guanylyl transferase enzyme. These enzymes are nothing but it is going to add a guanidine residue at the 5' end but in an inverted position. So it will only target the guanine residues and put it in the right inverted position of the 5' end. For example, if you have the cycling ring like this, it is going to invert the position and it is going to add into the 5' position of the group. So what happens here is, now a guanine residue has been added. So during this an energy is utilized. So GTP, that is guanosine triphosphate, is converted into GMP and other triphosphates is been released out. Pyrophosphate is released out. So you will have an addition of GMP, then you will have P, 3 P's. Already you have 2 P's here, followed by you will have a nitrogen base, then your nitrogen base, then you will have your RNA sequence, right? The final process of capping is known as methylation. So it is going to simply use S-adenosine methionine in the 7th position of your GMP. So first one is a triphosphatase, which is going to remove the name triphosphatase. It is going to add on the triphosphate group which is present on the 5' end. It is going to remove 1 phosphate group. Next comes your guanylyl transferase enzyme. This enzyme is going to add a guanine residue in the 5' end, but it is going to add in the inverted position. If you take the ring, it is going to invert the ring and then add it in the 5' position so that nothing can come and harm your 5, that is 5' end of your mRNA. Next enzyme which comes is S-adenine, that is methylation process, in which S-adenosine methionine that will go and add into the add a methionine in the 7th position of your GMP. So you will have a GMP in the 7th position of your name, a methionine will be added. So it is known as methylation process. So if you take your 5' to 3', it is going to add methylation. You already had GP, then the sequence is already with 2P, NP and then the RNA. So this capping, right, I'll put another color. This capping will protect the mRNA from exonuclease activity. Protect the mRNA from exonuclease activity. Second, it protects the mRNA from exonuclease activity. Second, it will guide this mRNA via nuclear pore to cytosol. So it is going to protect it from exonuclease activity and it will help you in the guiding of those molecules, that is a mRNA with the 5' cap, from the nucleus to via nuclear pore to the cytosol. So this is the first process of your post-transcriptional modification. Now we're going to go into the second process known as polyadenylation. So if you take a DNA template, your RNA is synthesized in 5' to 3' direction. So this is taken as codon molecules, which is gonna code for an anticodon and can convert it into a protein. But there are certain signals which is present at the 3' end of your mRNA molecule, that is newly synthesized mRNA, which is not gonna carry any codons, so it is not gonna disturb. So if you take this separately, you will have codons here, but here you will have, it is gonna recognize certain signal, and the signal is usually of a protein. For example, I am giving CSTF, CPSF and other complex proteins comes and recognize this sequence which has been present in the 3' end. And for example, drawing it again, 5' to 3'. So when an enzyme, so CSTF, CPSF, CSTF, CPSF, it is gonna make a complex here. So this complex, what happens is, you will have another enzyme, in a first enzyme which is gonna act on polyadenation is poly A polymerase. Poly A polymerase enzyme, along with, that is PAB you can call, that is protein adenine binding, that is called as poly A polymerase enzyme and poly A binding protein. So you have two, poly A polymerase enzyme which is gonna recognize this signal, and poly A binding protein is gonna add adenine residues at the end of the complex. So what happens is, it is gonna add enough adenines in the poly A tail of your newly synthesized mRNA. So once enough adenosine, that is adenines, are added, this poly A binding protein will dissociate. Once their function is finished, this protein which is capable of adding this adenine is dissociated. So this function will become, the enzyme's function will become null. So now you will have already, after undergoing capping, it has gone for adenylation. So already you will have a cap here. So I'm drawing it with a cap. So it is undergone capping. Now it is undergone tailing. So this is the second mechanism, and this is also gonna protect the, it is also got to protect the mRNA from exonuclease activity, because the lifespan of mRNA is very less, and it is gonna protect it from exonuclease activity. It is also gonna increase the stability. Still researchers are going on this. It is gonna increase the stability of the mRNA by protecting it. The third phenomena is known as the splicing. So today you're gonna see the process known as spliceosome mediated splicing. It's a protein mediated splicing. Under spliceosome, you will have different molecules U1, U2, U4, U5 and U6 proteins, which gonna help you in mediate the splicing process. So now I'm gonna draw how your mRNA will be there, which will be having exon. This is your coding sequence, exon 2 and exon 1. It will be bound by an intron. In the intron, you will have an adenine residue. This is known as the branch point, where two introns are joined together and exons are joined by an intron. Now, what happens is, this exon has to come in close proximity so that they can bound each other, and you're gonna have to remove this intron in such a way that these exons are joined. So for that, you will have usage of some that is spliceosome mediated protein molecules termed as U1 and U2. First you're going to see what is the role of U1 and U2. So I'm drawing it again, the exon. So you'll have a exon 1 and exon 2. So what happens is, now you'll have the adenine residue here. your uridine 1 is gonna act on, your uridine 1 is gonna act here, uridine 1, and here in this branch point and uridine 2 is gonna act. So what happens is, usually this contains uridine, like first one is this U2 will act on the U1 will act on the phosphate group, and U2 also will act on the adenine end. So they both will have a phosphate group, so it is gonna cleave the process. So in order to bring the folding process, what happens is, now this U2 and U1 this U2 and U1 spliceosome protein, which is present in exon and the adenine branch point, will undergo folding, will help you the exons in folding, because only once exons are folded, this can be removed and it can be joined. So it helps in folding to bring exons in close proximity, that is bringing it together. So next, if you look, the folding takes place exon 1, exon 2, and you'll have the intron with the branch point. Now what happens is, once the folding is taken place, the U1, U2 and the A will be removed. The U1 and the U2 will be removed. So next, the other group of spliceosome proteins are U4, U5 and U6. It's gonna act on the protein. So U4 is gonna come and see, these are the exon 1 and the exon 2. You will have a branch point that is made up of adenine. U1 and U2 are the spliceosome mediated splicing. Now for the folding to take place, 3 different types of like spliceosome mediated proteins are required: U4, U5 and U6. So what happens is, U4, U5 and U6 also goes and joins to the group. So once they join, for example, they are joining it here, so they're gonna join it here, U4, U5 and U6. So once the folding is taken place, the U1 and U4 it is gone away. So U1 and U4 is removed from the process. So the next what all you have is U2, U5 and U6. So U2, U5, U6, you can see the U2 here, U5 here and U6 here. So what happens is, this U2 will attack the phosphate end and cleave. U2 will attack the phosphate end and it is gonna cleave. So here you will have a phosphate end, it is gonna attack here and it is gonna cleave. So what happens, you will have an exon here. Because the phosphate attack, there is no thing here, and you will have a exon 2 molecule here, and here you will have a small piece of intron with the A, and also you will have U5 and U6, that is U5 and U6. Now what happens is, the U6 will have an hydroxyl attack on the U5. The U6 molecule with this exon 2 side, you will have a hydroxyl end which is gonna attack the U5. So U6 and U5 will be removed, then the exon 2s will be joined. Exon 1 and exon 2 will be joined together. So exon 1 and exon 2 will be joined together. So this is how splicing takes place. First, for splicing to take place, you will have spliceosome mediated splicing process. If you take an exon 1 and exon 2 are joined together by an adenine, that is an intron which is present as a branch point. So there are few spliceosome mediated proteins. First one is a U1 and U2. This will come and join to the exon molecules at a suitable position. Next comes U4, U5 and U6 and U4, U5, U6 proteins. This will help in the exons the folding. The phosphate and hydroxyl attack and the nucleophilic attack will take place within the exon molecule in such a way that these proteins and the adenine molecules will be removed and exons will be joined together.